Good day and welcome to our presentation. Today we will present our project on segmentation of seagrass blade images using deep learning. I am Isaac Vargas with my mentor Dr. Ruby Merubeulu from the Hyperspectral Optical Property Instrumentation Lab and Digital Data Processing Labs at the Department of Engineering at Texas A&M University Corpus Christi. Our co-authors are Mr. Chi Huang and Dr. Kurt Camarada from the Department of Life Sciences at Texas A&M University Corpus Christi. Dr. Kurt Camarada serves as the biology and seagrass expert in our group. Mr. Chi Huang has just recently graduated with his master's degree in marine biology. Here is the outline of the presentation. We will introduce the problem and discuss the motivation and project goal. We will highlight relevant previous work and background, then describe the methods used in this work, from previous sample collection, image acquisition, to deep learning algorithms for seagrass segmentation. We will discuss the results of the implementation of deep learning algorithms and compare the performance of two deep learning architectures. Finally, we will present conclusions. Seagrasses are important for the marine environment because they support marine life and contribute to the health of the environment. Seagrasses are known to be covered by myriad epiphyte species, other algae, as well as animals such as tube worms. Thus, they are also considered to be bioindicators since their surface cover and composition reflect the nutrient levels in their benefic environments. Seagrass surface cover and composition are important for biologists as this information can be directly related to nutrient levels and health of the benefic environments. We present the application of two deep learning algorithms, ResNet 18 and ResNet 50, and are used to achieve semantic segmentation of seagrass blades, a tool for seagrass surface classification and mapping. Using these two deep learning neural networks, we are interested in determining the percent cover of each composition of seagrass surface. In this study, we also compare the computation performance of two deep learning algorithms. For previous work, Roos and his group compared deep learning convolutional neural networks to traditional machine vision techniques. More specifically, they used the inception network and compared their results to the superpixeling algorithm, the simple linear iterative clustering algorithm. Their results demonstrate that the convolutional neural network outperforms traditional methods, whereas Weidman uses various deep learning networks such as the fully convolutional network and is used to get a prediction of the seafloor coverage of seagrass. In our work, we investigate the performance of residual networks, in particular with transfer learning. Both cited investigate seagrass cover and geographic area, whereas we investigate individual seagrass blades. The surface of seagrass could be covered by many different types of epiphytes, with different image attributes including color, shape, size, and texture. Some of these include filamentous algae, amorphous gray and brown algae, white and red crustose algae, other green algae, and tube worms. In this work, we group all algal and their epiphyte types into one group called general epiphytes, with the exception of filamentous algae. Our focus is on this classification of general epiphytes, filamentous algae, tube worms, seagrass blade cover, and background. The results and products for this work include segmentation and mapping of seagrass blade images into those five classes, followed by the computation and the percent cover of these five classes. Our method of semantically segmenting individual seagrass blade images is based on the deep learning ResNet 18 and the ResNet 50 neural networks. This slide demonstrates the ResNet 18 architecture. The input image is 224 by 224 pixels and passed through 18 layers and convolutional filters. Each rectangular prism block represents a layer in the network, and the arrows indicate the direction of the data flow. This is an 18-layer deep network where skip connections define this as a residual network. Most importantly for application, the last two layers are where transfer learning occurs. These are the fully connected and the softmax layers. All items prior to the last layers are learned features. Softmax is the class of probabilities that indicate items of highest probability. For semantic models, each pixel is labeled with a class or ID of highest probability. Fully connected and softmax layers are where the transfer learning occurs. All layers before are feature layers and remain unchanged during the transfer learning process. By modifying the softmax layer, the user has 100% probabilistic control over what the output can be. We expand from here with the Deep Lab version 3 algorithm for semantic segmentation. All algorithms are implemented and tested using MATLAB software tools. This is the architecture for ResNet 50. It is very similar to the previous, but now 50 layers deep. Note the multipliers of times 3, times 4, times 6, and another times 3, and the addition of 1 by 1 convolutions. With deep layers, there are many computations, and in order to lower the complexity, bottleneck architecture with 1 by 1 convolution is implemented. With ResNets, the idea is that the deeper and more complex the network, the better it can be at getting higher level features. We will use these for semantic segmentation on seagrass images and compare it to ResNet 18 from the previous slide. We will also analyze the computation time with performance. For imaging, seagrasses were sampled from 3 different sites around Corpus Christi, Texas. They were then scanned by a 1200 dot per inch high-resolution flatbed color scanner seen in the bottom left image. Scanning took place after the seagrass blades were placed in microscope slides and irrigated with water. This is the encoder-decoder ResNet 18 architecture of DeepLab version 3 Plus for semantic segmentation. DeepLab encoder takes the output of the learned ResNet features and applies atrus convolutions, which are the white outline shape. Atrus convolutions enlarge the field of view of filters. From there, the filters then concatenate, undergo pyramid pooling, convolve, then exit the encoder portion into the decoder. The data is then bilinearly upsampled by a factor of 4 and then concatenated with the corresponding low-level features from the ResNet backbone. These have the same spatial resolution and are concatenated, convolved once more to refined features, and then upsampled by 4, which makes it the same spatial resolution as the input image, and we get out our desired pixel-by-pixel semantic map. This algorithm was implemented through MATLAB with the shown function. This is similar to the previous with DeepLab version 3 for semantic segmentation. However, the backbone of the algorithm is now ResNet 50 instead of ResNet 18, implemented with the MATLAB function. Percent cover for each of the labeled pixels is determined by computing the ratio between the total number of pixels in each class to the total number of pixels representing a grass blade, which is the total pixel in the image minus the total pixels representing the background. Percent cover is calculated for each of 4 classes, which excludes background. Once the deep convolutional neural network are trained using transfer learning, each image is then analyzed using the following process. First, an image to be analyzed is selected, then the background is removed by converting these pixels to 0, leaving only the pixels representing the entire seagrass blade. The image then undergoes histogram normalization, and when the processing is complete, the image is resized, or rather divided into 224 by 224 image blocks to be entered into the ResNet deep learning convolutional network for classification and pixel labeling. Once the classification is complete, the percent cover for each of the 4 classes of general epiphyte, filamentous algae, tube worm, and the bare seagrass blade is computed using the ratio of pixels belonging to each class to the total number of pixels representing the seagrass. Background is identified as the 5th class, but in this it is a trivial case. The classification results are also used to generate a semantic map of the image, visually showing the locations of pixels that belong to each class. This slide represents the results of 5 sample image blocks, including the original background removed image, the ground truth map overlaid with the original sub-image, the ResNet 18 network classification results overlaid with the original image, and finally the ResNet 50 classification results overlaid with the original image. Ground truth is obtained through visual inspection and manual pixel labeling. Here you can clearly see the final segmentation map for each of the 3 classes, those being the ground truth, ResNet 18, and ResNet 50. This is without overlaying it on the original image. Similarly, this slide shows 5 other subsamples and results of classification and semantic mapping overlaid on the original sub-images. It is important to note that not all seagrasses were covered with all the classified epiphytes or tubeworms. This slide shows the final semantic map for the last 5 sub-images. The percent cover for the 4 categories of seagrass cover or composition is summarized in this table for the 5 sub-images presented in the previous slides. Overall classification accuracy based on ground truth are relatively similar for both ResNet 18 and ResNet 50. Similarly, this slide shows the seagrass blade cover for the 5 other seagrass images. Similar to the previous set, generally, the ResNet 50 deep network produced slightly better results in overall accuracy of surface cover computation with respect to the manually generated ground truth. On average, for the 10 tested sub-images shown in the 2 tables, the overall seagrass blade cover accuracy was 90.5% for ResNet 18 and 91.03% for ResNet 50. For computational performance, as expected, ResNet 50 runtime for each of the 224 by 224 sub-images were longer than the runtime for the ResNet 18 network. This is due to the number of deep layers in the 2 convolutional neural networks. On average, ResNet 50 runtime was about 75% more than that of ResNet 18 using the listed samples. We also note that the training time with transfer learning for ResNet 18 was 13 minutes and 31 seconds, whereas ResNet 50 was 19 minutes and 38 seconds. For the end goal, we want to apply this algorithm to whole images of seagrass. This slide shows the entire seagrass images for which the segmentation results are reported in the next slides. We do not have ground truth labels for these images due to their sheer size. However, instead of analyzing the accuracy with respect to the ground truth, we report the percent difference between ResNet 18 and ResNet 50 results. Also, the computational performance in terms of runtime is presented and compared for the 2 networks. This table reflects the percent cover for the entirety of seagrass blades, since there was no validation data for the entire seagrass blades due to the labor-intensive labeling. Here we present a comparison of and the difference in percent cover measurements by ResNet 18 and ResNet 50 networks on classifying the surface cover. This is the second set of 5 seagrass blade images. Difference in percent cover for ResNet 18 and ResNet 50 for the entirety of the seagrass blades differed by a cumulative average of about 2%. Similarly to the 224 by 224 block images, the cumulative ResNet 50 runtime for each of the entire seagrass images was longer than the runtime for ResNet 18. On average, ResNet 50 runtime was about 60% more than that of ResNet 18 using the listed samples. As has been shown, seagrass blade surface cover accuracy for the 4 classes were comparable for the 2 tested deep learning convolutional neural networks. Although the ResNet 50 results were generally better than ResNet 18 results, in our tested cases, in 8 out of 10 cases tested, the computation time for ResNet 50 was significantly higher as expected. The trade-off between accuracy and computation time will depend on the application and the number of images that need to be analyzed. So, when some accuracy can be compromised, ResNet 18 could produce satisfactory results in favor of faster runtime suitable for real-time applications and in case of large size images and large data sets. We are currently expanding our analysis to more seagrasses to potentially generate statistically significant results. In addition, the deep convolutional neural networks will be trained through transfer learning for additional classes. to separate the epiphytes and the algal types beyond the four classes presented. The results of this work are expected to contribute to the characterization and comparison of the benthic environments at different locations based on the results and surface cover of the seagrass images. This project has been supported in part by the McNair Scholars Program at Texas A&M University, Corpus Christi. This concludes our presentation. Please contact us for any questions or for collaborative efforts at the provided email addresses.